in drug discovery pipeline, we have two major components. One was target discovery. There is another component, target validation, which is used for validating the identified targets. Now, what are target discovery and what are target validation? So, target discovery basically it is based on our genetic information, and it is based on or while it is discovered, keeping in mind that what are different diseases which are related to genes. On the other hand, target validation, it is used once the target is identified, discovered. Now we want to see that this target or this drug will work with human beings also, or for the animals for which it was discovered. So target discovery is the initial step and validation is the after effect. So in discovery we will be focusing on multiple drugs, but target validation will shorten those discovered targets. So how it is done, or what are different requirements for target validation? So basically for target validation we want to see, or we want to look at a relationship between genes and diseases. So for example, we can say what is gene to disease correlation in some animal model. So correlation means that how they can be combined together. Is there some positive effect or some negative effect, that is called correlation. So based on this approach, there are two broad categories. Number one is forward genetics and the other one is reverse genetics. Forward genetics is a mechanism when we have some genetic information and we want to move towards phenotype information. कि हमारे पास genes की information है, based on that genes information we want to see that what would be the effect of our drugs on the phenotypes. On the other hand, reverse genetics is different, that we have phenotype information और phenotype के traits हमारे पास available हैं. Now we want to see that कि इन phenotype के traits का क्या effect हो रहा है, and we want to go reverse towards genes. Phenotypes हैं, drugs लेंगे और उसके बाद हम देखेंगे कि genes पे क्या effect हो रहा है. Commonly used information or which should be used for drug discovery is forward genetics. So target validation tools. Now there are different target validation tools available, out of which we have discussed 5 in this slide. So what is happening here is they will be helping us in assessing genetic association of our drugs with the disease. So what is the relationship or association between our targeted drug plus disease? So different tools such as antisense agents. So what is antisense technology? It is opposite of sense technology. For example, if we have some nucleotide sequence, we will just reverse it, so it will become antisense. And then different molecules are used, such as we said that antisense oligonucleotides are used. So concept between these molecules are, they are basically inhibitors. So inhibitors means that we want to identify, okay, that if there is some protein, then it will be producing some disease at later on stages. So instead of let that protein flourish, stop that protein at the initial levels. So if we stop that protein at the initial level, then there are good chances that this disease will not flourish and that protein will remain safe. So drugs are basically molecules which act on cellular proteins such as enzymes and receptors. So what happens is, or downstream of this technique is that it will block events at nuclear or ribosomal event. So that's what I said, that instead of let the gene or DNA make the protein, stop that DNA before development of a protein, so that disease can be prevented. So advantage is that protein will be it will stop to have some protein developed, and after the protein is developed, it is difficult to cure or prevent the disease. Then we have ribozymes. So ribozyme technology is basically based on RNA mechanism. So as I said, that previous approach, it was stopping translation mechanism. This ribozymes, they are stopping basically transcription mechanism, that we have RNA molecules. So what will happen, these RNA molecules will cleave or clear other small molecules or RNA molecules which are not useful for any organism. So I have given two examples here. One, we have hairpin ribozymes. This hairpin ribozyme, it consists of only three bases, which are GUC, and then we have hammerhead. Hammerhead has NUH, where H can be any HUC, AUC, and N can be any of those four bases which are part of ribo RNA molecule. So mode of delivery. What is mode of delivery and what are these deliveries? Options of RNA based molecules or drug discovery molecules can be two. Either they can be started within the organism. If they are starting from within the organism, then they are called endogenously oriented molecules. And if they are coming or originated from outside the part, then they are exogenously moderated parts. Then we have two other categories of these molecules or target validation compounds, which are peptide nucleic acids. These basically peptide nucleic acid, they block protein translation. And then we have transcription factors, such as an example given here is we have zinc finder proteins, which has a very high affinity to bind and correct regions of inflected DNA. Then we have gene knockouts. So in gene knockouts, what we want to find out? We want to find out the mutant's DNA. Mutant DNA basically which have some change mechanism. So if there is some mutant DNA, then we want to remove that DNA before that DNA is transcribed or translated. So we want to remove that one so that our organism lives or remains safe, and there are very few chances of disease.